Thank you, and thank everyone for attending. I know there's an exciting meeting next door with the FDA, but hopefully this will be interesting as well. So today I'm going to give you an overview of Carisma. We're working on engineered macrophages. We're going to be making a couple of forward-looking statements. And also, I'm just going to read a real quick statement. The presentation today will focus solely on Carisma's proprietary platform technology and development programs. I will not be discussing the company's definitive merger agreement with Sesen Bio, which was announced on September 21st. For more information on the merger announcement, including the merger announcement press release, please refer to the website and Sesen Bio's recent filings with the SEC. If there are any today, please limit questions to Carisma's science and programs. Okay, with that out of the way. So Carisma is, as I said, working on engineered macrophages. We have been around for about 5 years. We're a Penn spin-out. The idea was hatched in the Center for Cellular Immunotherapy that had a group that, of course, had a deep experience in CAR T. They saw an opportunity to go after a new cell type, the macrophage. And so what Carisma is I would say that Carisma is to macrophages what the pioneers before us, Novartis, Juno, Kite, are to T cells, and Faden and Carta are to NK cells. We're building out this platform, and we are going to drive forward through development and commercialization with a macrophage-based approach. To do so, we've set up a very robust platform with all the traditional adoptive cell therapy tools, looking at them with a macrophage-centric lens. We have a strong patent position, carving out our space in engineering macrophages for a variety of applications, and so we have composition of matter on that front. We have a lead program that is currently in a phase 1 clinical study. We're generating data. I'll talk a little bit more about that, but it's really setting the stage for proving safety, mechanism of action, and feasibility of engineered macrophages in patients. We recently announced a partnership with Moderna around an in vivo strategy. So we are currently an autologous-focused company. We are putting pieces in place for both the allogeneic and in vivo delivery. I think the important thing is, as we sit here today, there are multiple value inflection points for the company as we go forward. And I'll try to touch on each of those as we go forward. So why macrophages? Macrophages are a unique cell. They're a member of the innate immune system, quite distinct from the other cell therapy approaches. They are a tissue-resident cell. They're found throughout the body. You can find them in the liver of Kupffer cells. You can find them in the CNS as microglial cells. You find them in the tumor as tumor-associated macrophages. They serve a variety of functions in the body. They're generally known as the first responders, so they will have activity to identify things that shouldn't be there and eliminate them through phagocytosis. They generally orchestrate the immune response and sort of recruit and activate other effector cells. They're involved in a variety of disease processes, fibrosis, wound healing, things like that. But we're going to take, our approach takes advantage of the natural abilities of the macrophages, and then we harvest the cells, we engineer them, and put them back in the patient. The mechanism of action that we have is multi-modality. The cells are generally set up to address the most challenging problems in solid tumors. The ability to access the tumor, the ability to overcome an immunosuppressive tumor microenvironment, and antigen heterogeneity. So as we re-administer the cells, what we expect is that the cells will traffic to tumor. They travel along a chemokine gradient and accumulate preferentially in the tumor microenvironment. Once they arrive, they're unaffected by that immunosuppressive environment, and it starts their activity. First, through recognition of the tumor cell as something that should be removed, so it engages a cell surface antigen and triggers phagocytosis. So there's direct killing mechanisms through macrophages. Secondly, macrophages, we've polarized these cells to a pro-inflammatory phenotype, and so they'll be producing a variety of cytokines that warm the TME. So taking a cold, immunologically cold environment and converting it to a warm or hot environment. Macrophages also, as I said, recruit in other effector cells, so we see an influx of T cells, dendritic cells, et cetera. And then lastly, what is important is macrophages are professional antigen-presenting cells. And so as they're eating through the tumor, They'll process and present various secondary antigens out through MHC to the T cells that recruited in, leading to what's known as epitope spread. So not just the primary antigen that we've targeted, but additional secondary antigens that are for that patient specifically. So this is our proposed mechanism of action: preferential accumulation in the tumor, killing through phagocytosis, warming the TME, and generating an adaptive immune response through antigen presentation. Now, to accomplish this, of course, we had to build out our platform. And today we have a very broad platform that it really covers several things. Number one is the ability to engineer macrophages. Macrophages tend to be resistant to lentine AAV, and so we had to go through a pretty robust discovery process to find a vector that would work on macrophages. We identified the first one is Ad5F35. It's a good transduction efficiency, 80% plus. And that's really the vector that we started with. Not only does it introduce the gene, but it also has the added benefit of polarizing the cells towards a pro-inflammatory M1 phenotype. So we have established the vector. Number two, we found a second vector that we licensed from NYU that it enables us to create an M0. And then lastly, we looked at mRNA as another mechanism of gene delivery. So we have multiple ways to deliver the gene of interest. We can engineer macrophages, which are our ultimate effector cell, but we can also engineer monocytes. Monocytes are the precursor cell. They're both terminally differentiated, but monocytes are usually they traffic, and then when they become tissue resident, they become macrophages. We can now engineer both of those cell types. We have a variety of payloads that we can put into our cells, CARs and all the associated functions, but we can also add additional transgenes for enhanced cytokine production, things like that. So we just have a very broad platform that continues to grow as we progress, and really we can do a lot with these cells. And we have applications in solid tumors, but we can also start looking at other places that we're going. Importantly, as we look at the platform today, we're an autologous cell therapy company. We're learning a lot in our clinical study. We know what's working, what's not working, some deficiencies, where we need to go. But importantly, all the things that we're doing in the autologous platform inform the rest of the things that we're doing. So we are exploring allogeneic. Obviously, there's a market trend towards an allo approach to have consistency and off-the-shelf capabilities. We're working with the Blazer Lab at University of Minnesota to develop our allogeneic approach. In vivo, I mentioned a partnership with Moderna. This is a place that enables us to bypass ex vivo cell engineering altogether and have an in vivo approach, a genetic reprogramming of circulating monocytes and tissue-resident macrophages. All of that is under our discovery and development umbrella. And importantly, as we look at as solid tumors as our primary direction that we're going, everything we're learning there can be applied in the non-oncology space. And so we are looking at liver fibrosis. Macrophages will traffic to liver. We're looking at neurodegeneration, targeting those microglial cells that I mentioned earlier. And we're also looking at autoimmunity. So what we're learning today in the autologous section is being deployed elsewhere. So for example, liver fibrosis, we're looking at an allo approach. And neurodegeneration, we're looking at an in vivo approach. So we're leveraging the clinical learnings today across multiple modalities. With respect to the partnership with Moderna, it's a deal that we announced in January. It's a very robust deal, very important for Carisma. It opened the door to the in vivo approach, something that we didn't have before. We had been working with mRNA, but we were unable to, we didn't have a way to deliver that in vivo. So we set up a partnership, a broad partnership with Moderna. It's a 5-year partnership on 12 oncology targets. We received $45 million up front, $35 million equity investment, and full R&D funding for each of these 12 programs all the way through. On the back end, we have a very significant milestone and royalty as we eventually commercialize these programs. It's a promising area. We rapidly were able to demonstrate the ability to be myeloid cell tropic with our LNP. And so we have early data already showing that we can deliver our genes to monocytes and macrophages. We're moving quickly to our CAR. We can deliver our CAR, and we anticipate that by sometime in the first half of next year, we'll be able to have proof of concept data in a tumor model and leading to a potential clinical candidate nomination by the end of next year. If you look at our pipeline, maybe it doesn't show up particularly well here, but we have a number of shots on goal. We have the autologous approach in the blue lines in oncology. Our lead program, CT-0508, is in a phase 1 study. We will be moving that into a combination study in this quarter. And we also have a CAR monocyte program. We'll be filing an IND on that mid next year. We have a mesothelin targeted program and a PSMA targeted program. We also have some, as I said, a couple of things outside of oncology that we're moving through discovery at this stage. On the in vivo side, we have the 4 programs that have, from Moderna that have been nominated so far out of the 12 that were targeted. Those are moving forward, and again, we'll have proof of concept data next year. And then lastly, an in vivo strategy for neurodegeneration is on the mix as well. Now, the clinical program, I'm not going to go through a lot of data. I'm just going to highlight a couple of points. We presented this data at ASCO in June, and the important thing is that the phase 1 data to date supports our overall hypothesis. We've been able to demonstrate first the feasibility of making engineered macrophages. We have good CAR expression, we have good cell viability, and we are able to make between 1 and 5 billion cells from a single patient's apheresis. We are able to administer these cells to patients with no preconditioning regimen required, and we're seeing evidence of trafficking. On a preliminary safety basis, we're seeing a very clean safety profile, limited CRS. We predicted low CRS because cells leave circulation very rapidly. We saw a very clean safety profile. And importantly, we're seeing the mechanism of action occurring. We're seeing trafficking and evidence of the accumulation of our cells in tumor. We're seeing evidence of changing the tumor microenvironment to a warm phenotype and recruiting and activating other effector cells. We're seeing an influx of macrophages, additional bone marrow-derived macrophages. We're seeing an influx of T cells. And lastly, we're seeing evidence of an adaptive immune response. So all the mechanisms that we're expecting, we are seeing. What we don't have today is a response yet. These are heavily pretreated patients, and we anticipate that with greater patients coming on, that this mechanism will translate into response. Our strategy is multifold to ensure that that occurs. Number one, we're looking at combining with a PD-1 inhibitor, or a T-cell checkpoint inhibitor, anti-PD-1. We're starting that study this quarter, and we will be able to show preclinically, we've shown strong synergy between for the 2 compounds, the CT-0508 plus pembrolizumab, and so we're starting that study. Number 2, we're looking at the numbers of cells. As I said, we're able to make between 1 and 5 billion cells. We're going to be approaching a CAR monocyte strategy where we can make up to 10 billion cells. There are also some additional benefits with that approach in terms of trafficking and cytokine production. So we're excited about that. We'll file an IND in that program mid next year. Thirdly, we're looking at next generation CAR constructs. We have a very straightforward CAR with one domain CD3 zeta. We're looking to add into that, and we've developed over 200 CAR constructs, and we have an idea of the direction we're going to go in the future. And then lastly, we're looking at novel modalities, allogeneic and in vivo. So this is all part of our platform development strategy as we go forward. If we look at the milestones and sort of our operating plan, it really falls into 3 areas. Number 1, clinical development. We are completing our cohort 2 of our phase 1 study, where it's all cells on a single day versus over a week period. We have an intraperitoneal administration for regionally disseminated gynecologic cancers. We have our anti-PD-1 combination. So clinical development with our lead program, CT-0508. Number 2 is to continue to develop our platform. We're progressing our next generation CAR design, and we'll deploy that in our next product. We are developing CAR monocytes in an allogeneic approach as well, and those are moving forward. We'll have data throughout 2023. And we're developing our in vivo strategy as well, and we'll have a neurodegeneration program there. And then third is to advance our pipeline. So we have our CAR mono proof of concept will be soon. We'll file our IND for mesothelin by the end of, towards the end of next year, and we'll progress our neurodegeneration programs as well. So to summarize, Carisma is really creating a new cell therapy approach with the myeloid cells. We have a proprietary platform that has robust capabilities. We have a pipeline of emerging programs, CAR-M programs. We have a validating partnership with Moderna and clinical data validating our approach as well. We have an experienced leadership team that really has decades of experience in cell and gene therapy and oncology drug development expertise. And as I said, we have multiple value inflection points as we look forward and continue to develop this approach. I have 4 seconds left for questions. All right, thank you.